So recall that a B cell is produced in the bone marrow. That's where the B comes from. It also matures in the bone marrow. And then naive B cells, naive meaning they have not been activated yet. So naive B cells usually will wait in the lymph nodes to become activated. When the naive B cell becomes activated, usually through the help of a helper T cell, what will happen is the B cell will make many clones of itself. This is called clonal expansion. Okay, so clonal expansion occurs, the B cell replicates. From here, B cells will differentiate. Some will turn into what's called plasma cells, and some will become memory B cells that will remember the specific antigen. Plasma cells are the antibody producing factories. Okay, so that's where we're going to focus in here on these antibodies that are produced by the plasma cells. And plasma cells are the only cells that make these antibodies. So an antibody is a protein. So these are proteins, and they have this Y shape. And another term for antibody that you'll hear is immunoglobulin, or Ig. So here's what an antibody looks like. In the green here, the green part is the heavy chain. Okay, heavy because it's the bigger subunit. And the gray here is the light chain. What holds these two sections of the Y together right here is a disulfide bond. So disulfide bonds hold them together. The region from this line to the bottom here is the constant region. The constant region will determine the type or the class of immunoglobulin. And there's 5 classes of immunoglobulins. The top part here is the variable region, meaning each antibody, every single one, varies in only this top part. So let's talk about this antigen binding site, okay? The antigen is going to bind at the arms. So antigens bind arms of the Y. So there's 2 antigen binding sites in this particular antibody. This region, the constant region here, is the stem. It's going to serve as a red flag or a signal to let phagocytes know, hey, there's a problem here, I need your help, okay? So this is kind of like the red flag region. This top part where the antigen binds, the variable region, is often referred to as the Fab region, F-A-B, okay? So this region and this region, these are Fabs. The stem part is often referred to as the FC region. The part of the antigen that actually binds here at the arms, okay, is called an epitope. So an epitope is the segment of antigen, the bad guy, where the antibody binds. Typically, the epitope is going to be a kind of a small protein subunit. Often, only about 4 to 16 amino acids long. Okay, so the epitope itself is a very small part of something like a pili on the cell surface or a flagella on the cell surface. Okay, so it's a very small peptide portion of that. All right, so that's where, as you can see here, the antibody binds to that antigen. Now, like on one bacterial cell, you're going to have multiple antigens. So there's multiple different types of antibodies are going to bind throughout. And each antibody is specific to its epitope. I'll give you a very brief overview of each class. The two most important ones, I should really say the three most important ones are IgM, immunoglobulin G, and IgA. All right, so let's talk about IgM first. IgM is very large because it's a pentamer, meaning 5 antibodies will stick together. And I have a picture here. So down here at the bottom here, this is IgM. You can see 1, 2, 3, 4, 5. Okay, 5 different antibodies will come together and form IgM. It's a 5 unit immunoglobulin, which means because there's 5 of them, it means there's 10 antigen binding sites, because each arm is a binding site, so there's 2 on each. It's a big one. Because it's so big, it can't pass from a fetus, from a mother to a fetus, for example. So it's too large to pass most barriers. But what's key about it is that it's the first class of antibody made against a specific antigen. So if a plasma cell has been introduced to an antigen for the first time, IgM is the first class to be made against that antigen. IgG, on the other hand, it's pretty abundant within the body, but it's the second class made. Okay, so it's the second class to be made. It's smaller, okay, so therefore it can be transported from the placenta into the fetus's bloodstream. Okay, so it can pass those barriers. It's smaller. Let me show you an example here. So let's say this is a vaccine, or somebody's gotten sick with a disease and it's the first time. Okay, so the first injection of a new antigen, such as antigen A. At the first introduction of this antigen, IgM is the first class of antibody to be made. So you can see here within 7 days, IgM is made. But IgG antibodies are made about a week later in this example. Okay, so IgG fall behind IgM during the first injection. Now let's say a person is re-injected with the same antigen, or they get sick again with the same pathogen. What's going to happen this time is IgG is going to explode immediately. So it's the first to be made in a secondary response, whereas IgM now is the second to be made, and there's not as much of it. So IgM is the first response. IgG is important for secondary responses. Okay, IgA, this is the most abundant type of immunoglobulin, so the most abundant class. IgA is so important for mucosal immunity. What that means is IgA is found on lining the mucous membranes And remember, the mucous membranes is where most pathogens enter from. So IgA is really important for preventing pathogens in the mucous membrane area. IgA is also secreted in breast milk, and it's also secreted in tears and saliva, and any bodily fluids like that. IgA forms a dimer. So if you look at this picture here, here's IgA. You can see there's two Y's, one here and then another here. There's two of them that attach. IgD is very not abundant. It's like less than 1% in serum of immunoglobulins. And we think it's involved in development of immune responses. IgE is barely detectable in blood, and we think it's involved in maybe like a helminth disease, like a parasitic worm, or perhaps also involved in allergies. Okay, now, what happens when an antibody binds to an antigen? Okay, and there's 6 main responses of things that can happen. The first is called neutralization. This happens when an antibody either binds to a virus, okay, like in this picture, a bunch of antibodies have bound all over the virus, or an antibody binds all over to an exotoxin that's secreted by a bacteria. So in neutralization, antibodies block the binding sites of the virus or the toxin. So antibodies bind and block the binding sites. So now viral spikes can't attach to the host receptor because antibodies are blocking them. Or a toxin can't bind to the host because antibodies are blocking them. So that's what neutralization means. Second thing that can happen is called opsonization. In opsonization, antibodies will coat the pathogen, such as in this case, they're coating the bacteria, and this makes it easier for phagocytes to recognize and engulf. So this is easier for phagocytosis. Because remember, these stems here will signal for phagocytes to come in and engulf. Third thing that happens, if an antibody binds to a pathogen, it can activate the complement system, which is part of your innate immunity. Fourth, antibodies could bind to the flagella of a bacteria, and this prevents the flagella from working properly, so the bacteria are immobilized, they can't move. Number 5 is called cross-linking. And this is where one antibody, such as this one, binds to 2 bacteria, or 2 epitopes from 2 different antigens at the same time. And this creates this giant antibody pathogen clump. And again, this is going to make it easier for phagocytes to come in and engulf maybe all of this big clump at once instead of just one pathogen at a time. The last thing that could happen is antibody-dependent cellular cytotoxicity. This is abbreviated ADCC. What happens here is, let's say you have a cell, a human cell here, okay, so this is a host cell, but it's been infected with a virus or a bacteria here on the inside. Antibodies can coat all the way around the host cell. And when this happens, this signals for natural killer cells to come in. Natural killer cells will secrete cytokines, and this infected cell will know to kill itself by apoptosis in order to prevent the spread of this virus.